Hello, my name is Sam Kang, and this is my final presentation for the Bio 140L lab class. In this lab class, we explored one key topic. How does the presence of pollutants in aquatic environments in different bodies of water affect CYP1A gene expression in brown bullhead fish populations? My personal objective in this lab was to observe TCAC2, a fish gill tissue sample from a brown bullhead fish caught in the Trenton Channel. The main objective, however, of the lab was to observe the effects of pollutants, specifically in this case, benzoapyrene, a type of polycyclic aromatic hydrocarbons or PAHs, on brown bullhead fish populations CYP1A, CYP1A, expression in Trenton Channel, Ontario. This lab also mirrored a manuscript written by Dr. Williams and Dr. Hubberstay in which they explored liver tissue samples. However, in our case, we explored gill tissue samples. The main purpose of this was to show the difference of gene expression in different fish populations across both contaminated as well as uncontaminated bodies of water in the Great Lakes. Below this purpose section shows a map of different contaminated bodies of water in these Great Lakes, which includes Trenton Channel, where my sample was from. Not only this, it also shows a photo of a brown bullhead fish, the main type of fish observed in both the manuscript and this lab. In the middle section, there's a flowchart which shows different steps we took in this lab to conduct something called an STS page analysis method using the gill tissues provided. Using pre-provided, pre-treated, and pre-dissected fish gill tissues, we incubated and centrifuged them to conduct cell lysis. After recalibration and dilution, we provided a Bradford assay. And conducting the sample from the Bradford assay into further recalibration and dilution, we were able to use gel electrophoresis to conduct STS page analysis. The results speak for themselves. On the right-hand side of the graphical analysis, on the top graph, there is a negative trend line, which directly correlates an increasing PAH value, the x-axis, with a decreasing CYP1A expression value, the y-axis. While this trend line is not a very accurate numerical value, as these are expected values, not the exact provided values, this value, which shows a slope of negative 0.3731, has a negative sign, which clearly states that if an increasing PAH value, the CYP1A gene expression of these brown bullhead fish in these water samples decreased. In the chart below it, there are the examined value and the expected values of the PAH contamination as well as the CYP1A gene expression. As not only this, below that, there's a legend where 5 names of different bodies of water, both contaminated and uncontaminated, are listed. On the right-hand side, we've seconded the first chart's analysis as well as conclusions by clearly showing that Bell River, an uncontaminated body of water, has low PAH value, the blue graph, but an increasingly high CYP1A gene expression value, the red graph. On the most right-hand side, we see sample number 4 and 5, 4 being Trenton Channel, where my sample was from. Trenton Channel, being an extremely polluted environment, has a really high blue bar, which show, or rather, yes, the blue bar, which shows that it has a lot of PAH contamination. Comparatively, it has a much lower red bar compared to Bell River sample. The analysis that we can get with this is that the areas of more PAH value contamination results in their fish having their CYP1A gene expression decreased. Further conclusions can draw that brown bullhead fish living in these contaminated locations have had their gene expressions lower CYP1A values than their counterparts which live in these non-contamination locations, for example, Bell River. The next steps of this lab are containing 2 specifics. The first specific is potentially corroborating this data with other types of fish to fully solidify a general decrease in CYP1A, not only in the brown bullhead fish, but potentially biological fish similar in the anatomy and physiology of these bullheads, or even different, to completely show that the PAH value has a consistent negative trend and a negative decrease on the CYP1A gene expression. Not only this, comparing that the manuscript conducted liver tissue liver tissues in their SDS page method and we did gill tissues, another next step could potentially be using other tissues of the brown bullhead fish captured in Trenton Channel or other places to fully corroborate the data that CYP1A expression can decrease with PAH value in every single area of the fish's anatomy and physiology. The lab could also expand their research by looking at areas not only outside of the Great Lakes, but potentially more areas inside the Great Lakes to expand their data variables and expand where they get their data, and therefore make a much more accurate and much more reliable data set for the greater scientific community. Thank you for listening to my presentation, and I hope you have a great day.